All right, it looks like it's 8 p.m. GMT, so it's time to start our regular webinar on vector symbolic architectures and hyperdimensional computing. So today we are welcoming Ryan Calmus from University of Newcastle. So welcome, Ryan, and well, without any further delay, I leave the floor to you. So look forward to the talk.
Thanks very much. And bear with me just while I deal with the machinations of the PowerPoint. Okay, so hopefully everybody can see that now. Okay, that's it. So yeah, hi everybody. Thanks very much for coming, and thank you for inviting me here to talk. So yeah, my name's Ryan Calmus, and I'm a PhD student within the Laboratory of Comparative Neuropsychology at Newcastle University. My PI, Chris, is also here today, so thanks for coming, Chris. And yeah, anyone has more questions I won't be able to answer, Chris is your man. So yeah, so I have a somewhat non-linear background, having moved into cognitive and systems neuroscience by way of computer science, medicine, and neuroscience, really. So I'm currently writing up my thesis on intracranial signatures of structured sequence processing in the human brain. And this is a project that incorporates both electrophysiological analysis and computational modeling. And so that leads us to today's topic, really, which is sequence structure in the human brain, vector symbolic architectures as a neurocomputational lingua franca. And so in this talk, I'm going to focus on a neurocomputational account of sequence processing through the lens of hyperdimensional computing. If anyone has any questions, I'm happy to take them at the end, of course. But otherwise, hopefully there'll be something for everyone here. So I'm not going to talk in too much detail about the introductory topics, but certainly to motivate what I'm going to be talking about today, I just should give a little bit of background. So we still possess a fairly limited mechanistic understanding of the neural dynamics supporting sequence processing in the human brain. And an improved understanding of these interactions can inform neurocomputational models that explain how incoming sensory stimuli are ultimately transformed into complex mental structures, and vice versa. So such models have the potential to support the generation of highly specific hypotheses about the neural representations that support sequence processing and language, which can then be empirically tested. So the main aim of this presentation is to draw together the neuroscientific evidence and a number of key concepts on the subjects of vector symbolic model design to form a strong argument for the central thesis that at present, vector symbolic architectures are the most appropriate route to an holistic account of sequence processing in the human brain. So my colleagues and I set out a lot of evidence in support of this claim in our 2019 paper, which I'll link to at the end of this talk. So do check it out if you're interested in learning more, since I won't be able to cover everything here today. And in particular, you'll also notice that this slide here mentions model testing, which is somewhat beyond the remit of today's session, but it's definitely a very important topic and one that we've been investigating in our lab. So I'm happy to talk more about that in future with anyone who's interested. So vector symbolic architectures, I just very briefly introduce this because I know everyone here is familiar with this, but vector symbolic architectures, or VSAs, define mechanisms for representing and manipulating symbolic content stored in high-dimensional vectors, and the vector symbolic operations proposed here and used in this talk are based on holographic reduced representations as defined by Tony Plate, and probably very familiar to today's audience. So here on the left, there's a little infographic showing actual vectors of dimension 100 reshaped into squares and manipulated using the key operators. So within such a system, the HD distributed vector representations can be reversibly combined to create a compound representation that's similar to using superposition or different from using binding its constituents. So here, superposition is carried out by addition and binding by circular convolution, and vectors can be compared using pairwise correlation or the dot product. So by supporting combinatorial operations on vectors, VSAs can, again, as I'm sure you all know, act as a really important bridge between connectivist architectures, for example, artificial neural networks or the brain itself, and algorithms defined at the symbolic level. So yeah, let's just focus on a small cross-section of the evidence on the neurobiological plausibility of VSAs. So in terms of the vector representations themselves, there is evidence that both localist and sparse distributed representations are used by the brain. And VSAs conform to this because sparse distributed vectors exist on a representational continuum, which allows them to demonstrate characteristics of either localist or distributed encodings, depending on their sparsity. And in terms of vector comparison operations, which networks themselves would need to be able to undertake to themselves compare two representations, it should be said that the dot product can be computed simply by summing presynaptic potentials weighted by synaptic weights, which means that any neuron could, in theory, accomplish this. So that leaves a final key issue in terms of these kind of architectures, which is whether the VSA notion of binding is acceptable. So often raised is this question of whether the brain implements binding using temporal or spatial mechanisms. And I think it's fair to say this is somewhat of a false dichotomy, since conjunctive spatial coding and temporal synchrony or asynchrony can be really thought of as different perspectives on the same dynamic process. So here, as a simple illustration of this, we can conceive of two representations, A and B, shown on the right, whose strength is modulated by some dynamic influence over time. And we can see that if representations A and B are temporally coordinated, then the downstream combinatory code is instantiated in a dynamic system using VSA. So it can be seen that VSAs ought to be supplemented by dynamic approaches to appropriately model the brain. And in terms of dynamic operations, whilst vector symbolic architectures provide the modeler with the tools to manipulate the symbolic content at an algorithmic level, relatively unencumbered by the choice of the underlying substrate, be that actual vectors or abstract connectionist networks or spiking neural networks. To support the modeling of biologically plausible neurocomputation, these population coding operations must be supplemented by these dynamic mechanisms. So basically, you've got Nengo is a library that many people here will again be familiar with, a popular Python framework that provides one method of adding dynamics to the story using spiking neural networks. But you've also got at a more abstract level ways that you can simulate dynamics to investigate this aspect without the computational demands of spiking neural simulation. So shown here are just some key examples of this. The fact that oscillations can drive the timing of discrete operations, that anti-synchronous waveforms can be used to segregate operations in time, and likewise synchronous operations can be used to coordinate representations in time. And finally, a more specific notion of phase coding, where the phase of an oscillation can be used to drive downstream operations. So let's start by considering the basic notion of how a VSA can instantiate an abstract sequence, a topic that, again, I'm sure is quite familiar to many of you. So ordinal positional encoding is a method of encoding an ordered list that relies upon unique ordinal identifiers to access each item, and a heavily explored method of sequence representation in the cognitive literature. And so this is as opposed to storing items by chaining, for example, some list A, B, C being stored by storing the relationships A goes to B and B goes to C. And it's also in contrast to just discarding all the order information and storing an unordered set. So in VSA terms, that would look something like this, where list is the superposition of all of the position, item, binding, and pos and item are the key and item vectors respectively. So the use of orthogonal positional keys permits substantial recovery of the individual items by unbinding, which can then, of course, be cleaned up as well. So this is an appropriate way to store an ordered list of items, but is needlessly computationally limited and explanatorily over-constrained, really, as a model of sequence representation in the human brain. So a less constraining extension of this idea is to encode items by relative rather than absolute position. So relative serial position conceives of items encoded relative to finite boundaries, as shown here, where you can see that the position of the items is stretched and squashed with the position of the boundary. So these boundaries could be derived from incoming sensory information or from some high-level expectation. Using this scheme, the position of the fifth item in 10, for example, would be encoded using the same key value as the second item in 4, for example. So there's considerable evidence for the existence of relative position encoding in the human brain, and including within the hippocampus and in motor, premotor, and dorsolateral prefrontal cortices. So in that regard, shown here are the activation profiles for 3 motor cortex neurons reported by Adam Carpenter and others in their 2018 paper on the encoding of serial order in rhesus macaques during a memory scanning task. And crucially, these neurons appear to exhibit activity profiles encoding proportional or relative serial position, irrespective of the absolute length of the encoded sequence. So relative positional encodings inherently explain the increased likelihood of confusing adjacent elements in longer lists. It's the relative terms, they're closer together. And they also potentially permit the abstraction of ordering information over different sequence lengths, which could be a really useful feature when learning sequencing dependencies, that is relationships in sequences that you want to abstract over different types of sequences or lengths of sequences. However, unlike absolute ordinal position that I've just demonstrated on the previous slide, encoding relative position using a VSA requires us to define a reliable way of transforming real scalar values to distinct positional key vectors. So how do we do that? So let's talk about relative serial order encoding and how we can encode a continuous value. So the first way of transforming a scalar into a vector trajectory is by using fractional binding. So a continuous scalar input value, which we should call alpha here, can be transformed into a continuously varying vector symbol suitable for downstream binding or unbinding or whatever you want to do with it by fractionally binding a given symbolic vector k with itself. So if binding is analogous to multiplication, this is analogous to raising a number to a fractional exponent. And here it's equivalent to binding k with itself alpha times, where in this case alpha need not actually be an integer, despite the fact that obviously we might typically think of binding as being a discrete and indivisible process. This was proposed by Plate initially, where he described how this innovation could be used to encode a continuous trajectory by computing the following equation you can see there, v equals the inverse Fourier transform of the Fourier transformed original vector raised to the power. Alpha is just some continuous value, including any fractional value, and V is the resulting vector symbolic encoding or key vector that you want to use. So this can be used in other vector symbolic operations, as with any other vector, as I said. It's beyond the scope of this presentation to talk in detail about this particular method, but note that it's also been relatively recently explored along with implications for hippocampal grid cell encodings in 2 really great conference papers by Comer and others, and Dumont and Elias Smith, that are definitely worth checking out. However, this isn't the only way to turn a continuous scalar into a positional key. So a continuous input value can alternatively be transformed into a continuously varying vector symbol, suitable again for downstream binding, etc., by linearly combining a set of arbitrary distinct basis vectors that are each scaled by basis functions uniformly or non-uniformly spanning the input domain. Here shown as Gaussian basis functions spaced evenly so as to span the input domain in the diagram on the right. So each distinct vector is scaled by multiplying each of its components with the value of its associated basis function. And the scaled vectors are then superposed, which produces one key vector that varies continuously with the input scalar. So this key vector can serve as the input to a relative serial ordered organized list. So in plain English, this is like saying that a real valued scalar is transformed into a continuously changing vector by summing a mixture of vectors and using the scalar input to control the mixing proportion. Here, there's an arrow showing how at that point on the x-axis, you would end up with an equal mix roughly of the 2 vectors shown. So unlike the former method, this encoding system explicitly permits non-uniform spacing of basis functions or a mixture of basis functions so that the input space can be optimally mapped. So it's also thus less assumptive than the previous method in terms of the actual representations that you might expect to see. So shown here, for example, are the input values at which 8 Gaussian basis functions peak. And so if the line were diagonal, the functions would uniformly span the positional space. But here, the sigmoid curve means that the codes non-uniformly span the input domain so that the positions at the edges of the input domain are better covered than those in the middle. So on the right, we can see these positional codes derived from that particular sigmoid basis spacing being used to simulate a crude but extremely parsimonious model of primacy and recency effects in free recall of lists of items, all of which are represented using this relative serial order encoding using the non-uniform basis function distribution. And to obtain these results, the only additional thing needed was simulation of item decay. but there were no further buffers or rehearsal mechanisms or parameters required. So it might be very crude, but it is nevertheless a very parsimonious model for this particular phenomenon. As with plate, this can be extended from just encoding scalars, this method, to work with input domains of arbitrary dimensionality. So this would allow the same kinds of representations and mechanisms that one might use to encode, say, the position of a rat in a maze in 2D, to also be applied to, for example, remembering a telephone number in 1 dimension, time. So this kind of thinking is concordant with a lot of evidence on the continuum of space and time encodings in the hippocampus. And ultimately, this allows the hippocampus to potentially occupy a more generally useful and central role within a cognitive model. So let's talk a bit more about the hippocampus. For a hippocampal model to have a useful computational role within a broader model of human cognition, it must be able to flexibly coordinate with external systems. So oscillatory activity has a known role in local and long-range coordination within and between regions of the brain, and particularly in the hippocampus. So on the right, we can see the output of a simplified hippocampal model, which is capable of serving a useful role in a broader model of sequence processing. And this vector symbolic model not only produces simulated place cell activity that serves a functionally useful role in replaying a sequence or predicting upcoming elements, but it's also explicitly coordinated by theta oscillations. So this oscillatory control means the model is able to repeatedly sweep forward through a stored sequence starting from any point, much like future scanning behavior described in the neuroscientific literature. So how does it actually work? Here, a theta oscillation drives the repeated playback of a previously encoded sequence A, B, C, D. So the sequence has been encoded using the relative serial order encoding described previously. And to coordinate playback of the stored sequence, the estimated phase angle of the theta signal is extracted, to which a positional offset can be added if necessary to simulate movement through space or time. This creates a monotonic, repeatedly resetting signal that can be used to scan through our saved sequence. And from a neuroscientific perspective, the use of phase is also quite notable because this is going to result in coherence between the phase of the theta wave and the amplitude of downstream activity, which produces a phenomenon known as phase amplitude coupling, which is observed extensively throughout the brain, but also specifically within the hippocampus as theta gamma phase amplitude coupling. So in any case, this sawtooth shaped phase signal is then passed in as a scalar input to a positional key generator, employing either one of the aforementioned basis function or fractional binding methods. And the resulting positional key and the memory trace of the original sequence are ultimately passed as operands to an unbinding operation to extract the appropriate item. And this positional key can be visualized over time, as it has been here, by sampling a random subset of components of the vector, shown here by a very crude approximation as if they were firing rates of specific neurons sorted by onset. So the activity profile illustrates the fact that specific components of the key show both repeated sweeps and an overall trend through representational space, much like place cells during the traversal of spatial locations. And so finally, at the bottom, we can just about see a plot of the dot product similarity between the output of the model and each element of the sequence. And here, each element is played back in order from a simulated current position. And as our current position moves forward, we play back from progressively later points in the sequence. So A, B, C, D, B, C, D, C, D, D, nothing, for example. This is not only functionally useful, but it corresponds to the phenomenon of phase precession, which means if we look at the crude approximation of place cell activity, we see that the phase at which activation occurs advances relative to the ongoing theta oscillation, as observed in the true hippocampus. So oscillatory-driven vector trajectories can be useful functional mechanisms that have biologically plausible roles in computational models of sequence representation. From a neurocomputational perspective, though, these representations become more useful if we can describe exactly how they might be derived from incoming sensory encoding. So here, the mechanisms described previously are applied to incoming auditory stimuli, which follow some of the general, this model follows some of the general principles of a well-known conceptual model of speech processing proposed by Giraud and Popel in 2012. So in their conceptual model, they explained how neuronal oscillations may contribute to the packaging of speech into units at different temporal scales. And in this computational model, an incoming speech stream contains low and high frequency features that can be tracked by dedicated oscillators, as shown here. So for example, this is entraining to the low frequency content of the incoming speech. And as before, we can decode the phase of this oscillation and use it to drive a positional trajectory describing the order of all of the shorter high frequency units within the boundaries of lower frequency units. So for example, this is the order of the phonemes within a syllable or the order of syllables within a word. And the positions and items respectively are ultimately bound together and superposed to form a compressed representation of the recent speech stream as shown on the right. So, bear with me if the next slide loads. Yeah, so because these mechanisms are now clearly defined, we can take the conceptual model and implement it either as a dynamic DSA model using pure vectors or within a dynamic framework such as Nengo. And this is the looping video of the mechanism shown on the previous slide running in Nengo in Python, written in Python. So this implementation takes its sensory input from a neurobiologically plausible simulation of the auditory periphery, shown on the top left. And so here this model is listening to pre-recorded words spoken by a human adult. And on the bottom left, we can see the low frequency envelope being extracted. And then to the right of this, phase being extracted. So here the phase is extracted by a crude approximation of the Hilbert transform, which is produced basically by taking the original signal, delaying it, and forming a time series of complex numbers with the original signal as the real part and the delayed signal as the imaginary part. This can be computed in spiking neural terms, as you can see here in Nengo, but it can also be described in ideal terms. So the phase signal is then used to drive a positional encoding using the basis function method described earlier, and then this is finally bound using circular convolution to the neural septual coefficients for the currently playing sound, which corresponds to a representation of the recent features of the phonic item we wish to store. So thus far, we've looked at a neurobiologically plausible internal sequence representation and sought to plausibly describe its emergence from raw sensory input. But we can also take this representation further to hypothesize about the mechanistic role of the frontotemporal language network, which is a question which still poses major challenges for the neuroscientific community. So if the frontotemporal language network, the frontotemporal language network comprises a set of frontal and temporal regions, which are consistently engaged during the processing of language, I should say. And evidence shows also aspects of proto-linguistic sequence processing. So ventrolateral prefrontal cortex, for example, a region including Broca's area, marked here roughly, where it says areas 44, 45, has a vital but highly debated role in the processing of the syntax of language and in processing relationships between items and sequences, known as sequencing dependencies. But there's convergent evidence that different areas of ventrolateral prefrontal cortex contribute to the maintenance of different kinds of dependency representation. So here we can see an illustration of the ventrodorsal gradient model of sequence processing complexity in primates proposed by Wilson, Marsden-Wilson, and Petkov, which suggests that more ventral regions at the bottom, such as frontal operculum, are engaged during processing of elementary relationships where items are next to each other in a sequence, known as adjacent dependencies. And as we move dorsally, by which I mean upwards, networks are engaged in processing increasingly complex dependencies, including those between items separated by one or more items, known as non-adjacent dependencies, and even multiple non-adjacent dependencies that can be organized hierarchically. And this is important because it's long been posited that in humans, these hierarchical representations correspond to branching tree-like structures, and that an ability to manipulate these conceptual trees is critical to language. So a tree is readily constructed within a VSA by again extending our previous serial encoding. And the diagram here shows how incoming linear structures, which are difficult for a human to remember, can be recoded into tree structures, which are easier to remember due to their compressed organization. So here on the left, we can see how we might encode an adjacent or non-adjacent relationship by extracting the two important items from the sequence and encoding them as a new list using relative positional keys. So here, the area of prefrontal cortex known as the frontal operculum is theorized to have a role in directing attention to the salient items, which in this case are the A and B at either end of the sequence. And likewise on the right, we can see the same concept extended to represent a hierarchical relationship between two pairs of items. So the outer and inner pairs of items are encoded in the same way into compressed sequences, and then each of those compressed sequences is treated in turn as an item of a higher level compressed sequence. So Here we're still using either of the two relative positional coding schemes defined earlier, but at each level of embedding, we're using a different base vector or basis vectors to generate distinct positional keys. So this means this could all effectively be driven by oscillatory activity as previously described. It's also important to point out here that obviously the depth of nesting can't go on forever since the unbinding mechanism is lossy. And this is actually a very useful limitation because humans likewise can't understand sentences containing endless phrases nested within phrases, nested within phrases. So cognitively, that's quite plausible. The previous diagram, though, was, despite its complexity, not quite complete because for clarity, it didn't explicitly show the earlier stages in coding. So briefly, that's shown here along with the other stages. So essentially, the new stage is a buffer at the bottom of the diagram, and the rest is the same as shown on the previous slide. So evidence shows that humans can learn not just center-embedded dependencies like this, but also notionally more demanding dependencies, such as cross-dependencies, where important items are interleaved. So therefore, really, any model of dependency encoding should provide a mechanism that might explain this. And effectively, this gets around that, really. So by buffering a linear representation of incoming items, which are the bottom left 3 rows, and then subsequently transforming these into a nonlinear branching representation, which are the middle rows to the top of the diagram. This part of the model inherently supports the extraction of cross-adjacent or non-adjacent dependencies, because as long as it fits in the buffer, it can be extracted and then rearranged. So if we're implementing the mechanisms we have described, we can focus on adding dynamics to an ideal vector-based model or add dynamics by using a neural simulation framework like Nengo. And I'd just like to quickly draw attention to the fact that the supplementary code for our 2019 paper can be found on GitHub with the address here and the citation for the paper there, and includes what I hope some will find is a potentially useful tool for MATLAB VSA modeling to add to the existing set, and particularly expands this area, which involves MATLAB simulation of plates operators, but could be edited to incorporate other VSAs quite easily. So the crucial code is contained within a single file, and it uses MATLAB's class system to make VSA modeling as painless as possible, I hope. So here you can see how easy it is to compute plate superposition, binding, inversion, and comparison operations just by coding what look like normal arithmetic statements, for example, A times B, to calculate the convolutional binding between the two. Likewise, it's made pretty easy as well to plot the actual contents of the vector as well as itself, or also plot the results of comparison operations. So, and ultimately, the key principles outlined here or any other VSA system that you can conceive of really could be quickly prototyped using this approach. So if Nengo is considered, say, a neural compiler compatible with vector symbolic approaches, then this MATLAB library is essentially a rapid prototyping tool that can either stand alone or serve as a precursor to full spiking neural implementation. And I should point out that this code is under active development, so new features will be added in future. And one of the exciting things here is kind of having a little bit of a background in compiler design. I have actually added some potential pre-compilation steps here that enable you to effectively write out your model in MATLAB terms and then have it generate a Nengo description for you, which can then, of course, in turn generate the spiking neural model. So this could ultimately become quite a useful prototyping tool. So anyway, in conclusion, I mean, there's more that could be said about everything I've discussed here today, especially on the subjects of learning mechanisms and the methods by which we can test these models. But hopefully I've convincingly argued for the central thesis that vector symbolic architectures are a uniquely powerful common language for neurocomputational hypothesis generation and communication. And more specifically, that dynamic VSA simulation at both the latent vector and explicitly neural levels can assist in the triangulation of neuropsychological evidence and support the modeling of things like segmentation, salience filtering, relative serial ordering, codings of sequences, and chunking, and hierarchical structure building. So yeah, just like to briefly thank my supervisors, Chris Petkov, Yuki Kikuchi, and Ben Wilson for their support, as well as our lab funders. And yeah, I'm happy to take any questions now, but if anyone would like to know more about this or continue discussion beyond today's seminar, in addition to the mailing list, here's my email, and alternatively, do check out this link to our 2019 paper.
All right. Ryan, thank you very much for this exciting talk. So now I open the floor for questions. So please unmute yourself and speak up. Maybe while people are slowly pressing the unmute button.
So, I realized there was a lot packed into that. So,
no, but Ryan, I'm sorry if this question will be too naive, but I mean, I'm not a neuroscientist in any sense. But so you, when talking about plausibility and neurophysiological plausibility, like how brain computes dot product, for example, or it computes the similarity. So my question is, what is the fidelity of this computation? So if you have, for example, two sequences, so what is how fine-grained is the can be the comparison in the brain
between the model and reality?
Yeah.
Yeah, so that's a great question. So the level of the information that we usually obtain is at the level of local regions or very large networks. So obviously at that point you're looking at either voltages or blood oxygen levels if you're looking at fMRI, which reflects the population activity as a whole rather than the activation of individual neurons. And so, yeah, that's obviously a key difference there immediately between that level of representation and the sparse population level code that we would expect to be able to find. But of course, there are certain experiments that are undertaken where you can record from individual neurons or clusters of individual neurons that can reveal a lot about this. So there've been, for example, experiments that assessed the actual sparsity of representations in various areas like the hippocampus. And so I believe that there was an estimation of 5% sparsity, for example, there, which is a number that I often then use in the sparsity of my vector representations. So you can get an idea of specific qualities of the representations through that kind of study.
Mm-hmm.
But more excitingly, I think, yeah, so again, I'm not arguing for plates architecture specifically. I find it very useful. I also think that there are so hi Tony, but I also think that there are some there are just some key assumptions about the recombination of representations, which are really not that controversial when you look at the evidence. So for example, the idea that there are compound representations in the brain, which are conjunctive codings that reflect multiple separate representations being bound together. And that those representations can either look very similar to the constituents or they can look very different. That to me is not very controversial. And I just think as long as the modeling technique and the comparison methods we use stick to just those very basic tenets and don't go beyond that, I don't think that it's as controversial as some people outside of this field at least would probably think it is. But for me, one of the most exciting things is more recently, we've been able to look at how we might track specific types of information flowing through the brain in different directions. And that's quite promising because then you can say, for example, my VSA model suggests that we should have an ordinal encoding coming from this brain region and an item encoding coming from another brain region. Can we see convergence from those brain regions in the actual movement of information through the brain? So that's one way I would suggest that we can test these models, the flow of information. So, yeah. Sorry, that's a long answer.
Thank you. More questions?
Yeah, Ryan, very, very interesting stuff. Thank you for the talk. So yeah, I totally agree with you on sort of the high level view that you were expressing, that what particular VSA operation you use is of less importance than sort of thinking about how to combine and how things, they use dissimilarity or similarity.
Yeah.
And what I wanted to ask you about was one of the things that always sort of puzzled and disturbed me was, when I was looking at these, I was just using dense vector representations because they were convenient and worked well in computers. But
sure.
It seemed like, I don't know, I don't know that much about the neurophysiological work at all, but it always seemed like the brain used much sparser representations.
I think I would agree that in most areas of the brain that have been investigated in that way, it looks a lot like there are very sparse distributed representations being used. But I think it's by no means homogeneous. You know, it's there are definitely, and I mean there are definitely papers that are even arguing that there are localist representations in the brain that are not distributed at all. So again, the reason why I like PhET symbolic architectures, including yours, is the great feature that you can tune that sparsity. You can go from having an incredibly dense representation. If I have all the way down, to a very localist representation. So if I have a vector of dimension 1000 and I have a sparsity of 0.001, right, then I will only ever have one active component in that vector at once. And effectively, that at that point is behaving like a localist encoding. So I think that I definitely would argue that on the whole, these representations that I'm definitely interested in the frontal cortex are incredibly sparse representations. But yeah, I don't know whether it really answers your question. I just think they're powerful enough because you can tune that parameter. That's my opinion.
Yeah. So the question that I wanted to ask you was whether you'd seen any indication. So there could be two reasons for having sparse representations. One is that just the hardware works better with sparse representations. The other reason could be that there's some actual property of the representations that it can work better when the representations are sparse. And so I was wondering whether you'd seen any sort of evidence or indications along those lines.
I mean, there's a great paper by Ding and Popel, which actually set me on this path in the first place. So their paper was investigating how, and again, my focus is on a particular area of the brain, really, which is mainly inferior frontal gyrus, so frontal cortex. But they were investigating how humans track the opening and closing of phrases in speech. And one of the things their electrophysiological evidence suggested was that as you move through the representation of a sequence, the electrophysiological changes reflect potentially superposition of representations on top of each other. Now, to me, that's quite compelling evidence that these representations must be quite sparse to observe an increase in the actual population level electrophysiological signals, because if everything was dense, you would expect, not to be able to observe this increasing summation as obviously as they were able to. So that's my answer to that. At least in the area I'm looking at, I've not seen evidence that suggests that dense representations would be better. And indeed, I'm a bit cautious about this because I saw a paper recently that suggested that there's definite evidence against dense representations in the human brain. But I think some people might argue for different regions implementing that. But definitely frontal cortex, I don't think has much need of that. That's probably a really unnecessarily long answer for that. Sorry.
Okay, thank you.
Thanks.
More questions?
Ryan, it's Dennis speaking.
Hey, hello.
You mentioned that at the beginning of your talk that you might also use VSAs not only in the design of representations but in the testing. Could you say a couple of words on that? This sounds intriguing.
Yeah. So there are a couple of ways to do this. So firstly, I mean, when we were writing the 2019 paper, we often found that even just the step of formalizing these into algebraic descriptions of representations was incredibly useful in and of itself for creating testable hypotheses, which obviously down the line you can assess. And those are highly specific to whichever region you're looking at. But so in terms of like the life cycle of hypothesis generation and testing, I feel like VSAs are quite useful there because you can actually get to the point where you create new hypotheses just from the design of your model immediately. But alternatively, obviously, you can take this all the way through to doing something like a Nengo-based spiking neural simulation. And then you could directly compare the spiking neural output with real neural output through a variety of methods involving correlation, effectively. But what I've been focusing on is simulating only at the ideal mathematical level, but over time using dynamics, as you saw in the toy hippocampal model, for example. And so there's no spiking neural simulation there, but there are VSA operations and there is dynamics. The focus I've been using there for testing these representations against real life representations uses a multivariate comparison approach, which was first proposed by Kriegerskorter in 2015, called representational similarity analysis. I don't know if any of you have had to use this, but basically what it's known as RSA. What RSA essentially does is it asks you to characterize each system that you're interested in terms of the differences between the system's behavior under different conditions. So you end up with a map of these differences for, say, a neural system, and you can do the same thing for a neural system recorded with a different modality. You can do the same thing with neural systems recorded in 2 different species, or indeed you can do the same thing by characterizing the differences under different conditions for your model and doing the same thing for the brain activity. So what I've been doing is I've been using vector symbolic architectures to derive the predictions from the model, sticking that into representational similarity analysis, and then using that to test the model. There are a number of different ways of doing this, though, and but that's in preparation at the moment. That's what we're working on right now. So yeah, that's probably more than anyone ever wanted to know about that.
Thanks.
All right, we have time for maybe one more question, if there are any. Well, if not, please remember that we have our Google group, so you can post your question there. So I will add Ryan and Chris to the group, so that we can keep communication going on. All right. If there are no more questions to Ryan, I would like to thank you once again, and just to give you some statistics, at the peak of attendance, we had 21 participants, so just really good. I mean, it's kind of our normal attendance ratio. So thank you once again, Ryan, and yeah, thank you all for attending. So I see you next time in 2 weeks. And so this recording will be available online as usually. Okay. Thank you all.
Thank you.
Bye.
Thank you very much. Bye bye. Have a nice day.
Yeah, you too. Bye.